Welcome student on another video lecture. Today's topic is transferosomes. The term transferosomes means carrying body. What are these? These are the carrying bodies, and it is derived from Latin term transferi, meaning to carry across. It will carry the drug molecule across the membrane, that is carrying body, and from the Greek term soma, meaning body. Transferosomes are artificial vesicles resembling the natural cell vesicle. So these are the artificial vesicles and which are look like the natural cell vesicle. Thus are suitable for targeted and controlled drug delivery. So it can be used for targeted drug delivery of the drug as well as controlled release of drug for prolonged period of time. These transferosomes can be used. Transferosomes are vesicular system made up of phospholipid as the main ingredient with 10 to 25% surfactant. So it is made up of phospholipid and main ingredient in that is surfactant such as sodium chlorate and 3 to 10% ethanol. The surfactant work as a edge activator. What is the role of surfactant? Surfactant will act as edge activator and impart ultra deformability to transferosome to help them to squeeze through the pores in stratum corneum. So it will helpful to pass through the pores into the stratum corneum. Advantages: they can deform and pass through the narrow constriction. So it can pass through narrow constriction from 5 to 10 times less than their own diameter without measurable loss. They have high entrapment efficiency in case of lipophilic drug, near to 90%. Their high deformability allow better penetration of intact vesicles. They can act as a carrier for low and high molecular weight drugs. So both low and high molecular weight drugs can be passed, or they will act as a carrier for transfer of both high and low molecular weight drugs. Example: analgesic, anesthetic, corticosteroid, sex hormones, anti-cancer, insulin, gap injection protein and albumin. All these can pass through the membrane. They will act as a carrier for these drugs also. They are made up of hydrophobic and hydrophilic moieties. They can accommodate drug molecule with wide range of solubility. So these are having both hydrophobic as well as hydrophilic moieties, and they can accommodate the drug molecule with wide range of solubility. They act as a depot and release their content at low and steady rate. They can be used for systemic as well as topical drug delivery. So both systemic drug delivery as well as topical drug delivery can be possible by using these transferosomes. They are biocompatible and biodegradable as they consist of natural lipids. So these are made up of natural phospholipid, and due to which they are biocompatible and biodegradable. They protect the encapsulated drug from metabolic degradation. They are easy to scale up as procedure is simple and short, and to avoid unnecessary use of pharmaceutical unacceptable additives. Now, method of preparation. Here there are three methods mentioned for preparation of transferosome: thin film hydration method, modified hand shaking method, lipid film hydration technique. Here it is the structure of transferosome. You can see here this one is the hydrophilic drug, this is hydrophobic drug, this one is edge activator, and these are the phospholipid bilayer. So both hydrophobic as well as hydrophilic drugs are incorporated in this transferosome. Some examples of herbal transferosomes, which include capsicum annum is the biological source. Category is resin. Application is it increases skin penetration. Treatment of rheumatism, and the active ingredient is capsaicin. Curcuma longa, the category is resin. Increase skin permeability. Anti-inflammatory is the use, and curcumin is the active ingredient. Catharanthus roseus, category is indole alkaloid. It increases permeability. Use is an anti-cancer, and the active ingredient is vincristine. Colchicum autumnale, amino alkaloid is the category. Reduces GIT effect. Treatment of gout, and colchicine is the active ingredient which is present in the drug. Now, thin film hydration method. First step is mixture of phospholipid and surfactant. It is dissolved in organic solvent. So in case of thin film hydration method, first step is phospholipid and surfactant. They are dissolved in organic solvent, and here there will be incorporation of lipophilic drug is done. Thin film formation using rotary evaporator. Now the thin film formation is done by evaporation of solvent by using rotary evaporator. Place under the vacuum for 12 hours. Hydrate under buffer. pH 6.5 is maintained and the speed is 60 rpm. Here there is incorporation of hydrophilic drug is done. Then sonicate for 30 seconds. Homogenize. Extrusion 10 times through sandwich of 200 and 100 nanometer polycarbonate membrane. So first step is phospholipid surfactant, that is dissolved in organic solvent. At that time there will be incorporation of lipophilic drug. Then thin film formation by evaporation of solvent using rotary evaporator. Then place under the vacuum for 12 hours. Now hydrate that film which is formed or adhere to the wall of the flask by using buffer, and the pH is maintained 6.5. After that sonicate it for, and at that time there will be incorporation of hydrophilic drug. Now sonicate it for 30 seconds, and final step is homogenization and extrusion through polycarbonate membrane, and there will be result in formation of transferosome. This is the thin film hydration method which is used to prepare transferosomes. Now another method is modified handshaking method. In this method, first step is, this method will share the principle of both rotary evaporation and sonication technique. First step is dissolve lipophilic drug, phospholipid and H activator in organic solvent to produce a clear solution. So lipophilic drug, phospholipid, H activator, all these are dissolved in organic solvent to produce a clear solution. Then solvent is evaporated via handshaking. While the flask is partially immersed in heated water bath and the temperature maintained is 40 to 60 degrees Celsius. So that flask is placed in a temperature of 40 to 60 degrees Celsius on heated water bath, and hand shaking is done continuously so that the solvent is get evaporated. Now this process form a thin film along the inner walls of the flask overnight to allow complete solvent evaporation, and placed or kept it for overnight so that there will be complete evaporation of solvent and thin film is produced inside the wall of the flask. The film is hydrated with an appropriate buffer solution now and incorporating the hydrophilic drug with gentle shaking. Now at that step there will be hydration by using buffer solution and incorporation of hydrophilic drug. This will result into formation of transferosome, which is prepared by modified handshaking method. Another method is lipid film hydration method. In this method, lipophilic drug, organic solvent, age activator and phospholipid, they are taken in flask and they are mixed with each other, or complete dissolution is done to form a clear solution. Then the solvent is removed through handshaking by evaporation method. The flask is partially submerged in heated water bath, leading to the formation of lipid coating on the flask inner wall. Now hydrating that film with buffer to form vesicle. So this is lipid film hydration method. So these are the three methods we have seen, that is thin film hydration method, modified handshaking method and lipid film hydration method. These are used to prepare transferosome. So this is about the detailed account on transferosome. Thank you.